Question number 96. While analyzing the DNA of an organism, different proportions of bases were obtained, as given below. Adenine 28%, guanine 22%, thymine 28%, cytosine again 22%. From the above information, it can be generalized that first option, it is a single-stranded DNA. Second, it is a double-stranded DNA and follows Chargaff's rule. Third, it is a double-stranded DNA but does not follow Chargaff's rule. Fourth, it is a single-stranded RNA. Okay. By analyzing the different proportions of bases in the DNA, you can conclude that adenine and guanine both are purines. Thymine and cytosine both are pyrimidines. And you can see here that the amount of purine is equal to the amount of pyrimidine. And if the amount of purine is equal to the pyrimidine, then this DNA follows the Chargaff's rule, and it's a double-stranded DNA because Chargaff's rules is only for the double-stranded DNA. So the correct option for the given question is option number 2. Let's see the next question. Question number 97. If the sequence of nitrogenous bases of the coding strand of DNA in a transcription unit is 5' ATGGCC AT3', then the sequence of bases in its RNA transcript would be. If you remember that there are two strands in the DNA. It is 3' to 5'. It is 5' to 3'. The strand which codes for mRNA is called non-coding strand, whereas the opposite strand which does not code for mRNA is called coding strand. So this codes for mRNA. Now, if the sequence of the coding strand is given as ATGGCCAT, if the sequence of the coding strand is given, then you can find out the sequence of the non-coding strand which codes for mRNA, and it is T A C C G G T A, and as it codes for the mRNA, the complementary sequence of the non-coding strand, it will be A U G G C C A and U. So this should be the sequence of the mRNA. Now you can analyze one thing. See here, the strand which is the coding strand, the sequence of the coding strand and the mRNA, it is the exactly same, except at the place of T it has U. So you can remember this thing, that if the sequence of the coding strand is given, then the sequence of the mRNA will be exactly same to the sequence of the coding strand, except at the place of T you have to put U. It is very easy. Now you can analyze. It is ATGGCCAT and it is AUGGCCAU. Both are exactly same. So the correct option for the given question is option number 3. Let's move to the next question. Question number 98. If the anticodon sequence in tRNA is 3' AAA 5', the amino acid carried by it is first option serine, second valine, third phenylalanine, fourth glutamic acid. If the anticodon sequence in tRNA is this, then the sequence of the codon will be UUU, and UUU codes for phenylalanine. So the correct option for the given question is option number 3. It was a very easy question and the memory based. Question number 99. The lac operon of E. coli gets switched on when? First option, repressor binds to the operator. Second, lactose binds to the RNA polymerase. Third, lactose binds to the repressor protein. Fourth, inducer binds to the operator. What is lac operon? Lac operon was discovered by Jacob and Monod in E. coli, and it is a system which is related to the metabolism of the lactose in E. coli. So what happens when this lac operon is switched on? It means when transcription of structural genes takes place. So when transcription is on. Transcription can be on when this RNA polymerase binds to the promoter. When can this RNA polymerase bind to the promoter? Just you have to find out. The I gene, which is a constitutive gene, it synthesizes repressor protein. Repressor proteins binds to the operator, and then there is no synthesis of the enzymes. So this time this lac operon is said to be switched off. So you have to control this repressor anyhow. So when lactose is present in the medium, this lactose binds to the repressor molecule, and then this repressor cannot bind to the operator, and this time this RNA polymerase binds to the promoter and transcription takes place. So this lac operon is called switched on operon. So when can this lac operon can be switched on? When repressor binds to the operator? No, it's a switched off condition. Lactose binds to the RNA polymerase. No, lactose cannot bind to the RNA polymerase. Third, lactose binds to the repressor protein. Yes, when lactose binds to the repressor protein cannot bind to the operator. Hence, RNA polymerase can bind to the promoter easily and transcription takes place. Fourth, inducer binds to the operator. No, lactose is inducer and it cannot bind to the operator. So the correct option for the given question is option number 3. Let's see the next one. Question number 100. Mark the wrong statement. First option, in prokaryotes, transcription and translation occur in the different compartments. Second, in eukaryotes, usually structural gene of transcription unit is monocistronic. Third, in prokaryotes, the mRNA is produced in the cytoplasm. Fourth, in eukaryotes, the primary transcript undergoes splicing. So you have to mark the wrong statement. See, second, in eukaryotes, usually the structural gene of transcription unit is monocistronic. Yes, in eukaryotes, a single gene codes for a single cistron or a single polypeptide. So this is true. Whereas in prokaryotes, the structural gene is polycistronic. A single gene can code for many polypeptides or for many cistrons. Third one, in prokaryotes, the mRNA is produced in the cytoplasm. Yes, it is also true because transcription takes place in the cytoplasm. Hence, mRNA formation takes place in the cytoplasm. Fourth, in eukaryotes, the primary transcript, which is hnRNA, heterogeneous nuclear RNA, undergoes splicing. Yes, because hnRNA contains both exons and introns. Introns are the non-coding regions and the exons are the coding regions. So before translation, we have to remove introns from the hnRNA, which is done by splicing. So in eukaryotes, primary transcript undergoes splicing before translation. So this is true. See the first statement, in prokaryotes, transcription and translation occur in the different compartments. It is absolutely wrong. Why? Because in prokaryotes, there is coupled transcription and translation. These both processes take place in the same compartment, so they do not take place in different compartments. So the wrong statement is the first one. So the correct option for the given question is option number 1. Let's move on to the next question.